Hello, I'm Miles Nunn. I'm the Chief Scientific Officer at Akari Therapeutics. Today I'm going to talk about the science behind Nomacopan. Akari's drugs have an unusual source, ticks, which are blood-feeding parasites. When the ticks feed, they need to control the host immune response, and they need to control pain and itch and blood flow to prevent detection by the host and being groomed away. They do so by secreting a variety of proteins from their saliva. And what we've done is we've mined those proteins to understand how they interact with the immune system and to get them into a recombinant form so we can develop them further into drugs and then select which proteins we want to take forward into drug development. Our lead drug Nomacopan was selected because it has highly desirable pharmaceutical properties. It has a unique mode of action binding to C5 and LTB4. It's incredibly soluble, which means you can administer it via a variety of routes, subcutaneously, intravenously, as eye drops, or to the lung, and we're working on that program. It's very specific, which means it has fewer off-target effects. It's a small protein, it's about 9 times smaller than a monoclonal antibody, and that means it may have really good tissue penetration, which is going to be important in many of the disease states that we may choose to target. It expresses very well in bacteria, and in recombinant form we get an identical protein to that produced by the tick. So how does Nomacopan actually work? Nomacopan binds to C5 and LTB4, as I've mentioned. It locks C5 in a conformation, which means it can't be activated by the complement convertase of the complement system to generate the terminal complement complex, and it sequesters leukotriene B4 within the body of the protein. C5 and LTB4 both act by recruiting cells to inflammatory sites, where those cells then degranulate and produce cytokines, for example TNF, IL-17, and a variety of other pro-inflammatory mediators. Therefore, by inhibiting both C5 and LTB4, Nomacopan has a broad and potent anti-inflammatory effect. Importantly, C5 and LTB4 are both dysregulated in many disease states. This means that Nomacopan has the potential to treat diseases where those mediators are dysregulated. No other biopharmaceutical drugs have these unique properties of inhibition of C5 and LTB4 simultaneously. However, drugs have been developed which inhibit C5, such as eculizumab and ravulizumab by Alexion, which have been highly successful drugs. And large pharmaceutical companies have been very interested in developing leukotriene B4 inhibitors. The approach is taken historically as small molecules, and many of them have had off-target effects. We think Nomacopan may overcome some of the difficulties in leukotriene B4 inhibition that have been encountered in the past. Given its broad inhibitory activity and ability to be engineered, Nomacopan can be considered a platform molecule. We have preclinical data in a variety of disease conditions, including neurological, hematological, heart disease, in the eye and in the lung, and in a variety of other conditions, and there's more information on the website, which show you that Nomacopan is efficacious in all of these disease states and may therefore be used as a drug to treat these diseases in man. In terms of its engineerability, we know a fantastic amount about Nomacopan, having derived crystal structures and worked on the molecule for many years, which means that we can engineer it in really interesting ways. So we have variants that have a longer half-life, we have variants that bind to only leukotriene B4, we could make variants that only bind to C5, and we could target Nomacopan to particular tissues. All of these extend the number of diseases that Nomacopan could be targeted to, and also increase the intellectual property protection around Nomacopan. So what have we learned from our clinical programs that support the underlying scientific rationale? Well, first and most importantly, we found that Nomacopan is well tolerated in human subjects. That's not something we knew before we started our clinical programs. So we have 35 years of patient data, and the drug's been extremely well tolerated with only one possibly drug-related serious adverse event, which was a urinary tract infection. Secondly, we've found that Nomacopan maintains its drug effect. So patients who have been self-administering the drug for periods of up to several years have had their terminal complement activity fully controlled at all time points. Most importantly, Nomacopan is efficacious in patients, generating a strong clinical response. So our best data is in PNH patients, of whom 19 have been treated. 15 of those patients were transfusion dependent before starting on Nomacopan, and 80% of them became transfusion independent while on Nomacopan. Given these findings on tolerability, maintenance of drug effect, ability to self-administer the drug over the long term, and the efficacy of the drug, we believe the further clinical development path of Nomacopan is de-risked. To make the most of Nomacopan's unique properties of LTB4 and C5 binding, we're focusing on diseases where LTB4 and C5 both drive the disease pathology. The focus of our current clinical programs are BP, bullous pemphigoid, and TMA-HSCT, thrombotic microangiopathy after bone marrow transplant, and we have preclinical programs in lung and in the eye. If you'd like further information, please look at our website. Thanks very much.